everybody. My name is Hannah Dawson, and I'm a graduate student at University of Washington in the School of Oceanography. And today I want to talk to you about using metabolomics as a method to probe the physiological adaptations of sea ice algae. So just to orient us to this extreme environment, this is a video showing you the sea ice extent in the Arctic over one season. So sea ice is an extreme and dynamic environment here on Earth, with 4 to 6% of the global ocean area being covered by sea ice depending on the time of the year. And despite being a relatively harsh environment, a number of organisms have been able to exploit this habitat. And one of these organisms are sea ice algae, which are single-celled eukaryotic photosynthetic organisms that can live within the liquid brine channels formed in sea ice surrounded by a freshwater ice matrix. So here I'm just showing you an example of sea ice diatoms inside of a brine channel. But these organisms contend with a number of challenging features in this environment, and temperature is one of the largest challenges in this environment, where throughout the sea ice column, temperature varies greatly on a spatial and seasonal scale. So temperature at the top of the ice column is much colder and in contact with the atmosphere, while temperatures at the bottom are much warmer and closer to seawater conditions. And like I mentioned, there's a liquid brine volume left behind when seawater freezes, and that volume also scales with temperature. And lastly, salinity varies throughout the ice column also with temperature, with salinities of that brine being very high at the top of the ice where it's very cold, and closer to seawater salinities towards the bottom of the ice. And this is also a variable environment on a seasonal scale, with the melting of sea ice in the spring and reformation in the autumn and winter, bringing along large swings in temperature and salinity, reaching near freshwater conditions in melt season and temperatures above zero. But despite this, sea ice algae have been able to survive and thrive in this environment. And here I'm showing you a video of what it looks like underneath the sea ice in the Arctic in Utqiagvik, Alaska. And here we just stuck a GoPro down through a core hole in the ice. And I think this is a really cool video. It just gives us a glimpse at this otherworldly environment where these organisms are, you can see a brown layer on the bottom of that ice, and those are all sea ice algae, mostly dominated by diatoms. And in this environment, they can reach these high abundances like we're seeing here, high rates of primary production, and serve as an important food source for a number of organisms, and play largely into biogeochemical cycling and climate active gas production in this environment. But we still don't have a very good understanding of how they're adapted to do this. So I'm interested in how sea ice algae are physiologically adapted to survive and thrive in the extreme and variable conditions found in sea ice, and how we can probe those adaptations. So one way we can do this is by better understanding the metabolic reactions going on inside of a cell, or the suite of biochemical reactions that allow a cell to be alive. And this is just a glimpse at how complicated these systems can be. These are all the metabolic pathways in an example diatom cell, where each line in this diagram is a metabolic reaction mediated by enzymes. And along each of these lines, there are a lot of precursors, intermediates, and products of these reactions, which I'll refer to as metabolites. And one way that we can get a better idea of what metabolites are inside of a cell, how they're regulated, is by using a method called metabolomics. So metabolomics, we take this entire intracellular pool of metabolites inside of a cell, extract them, and then use a tandem liquid chromatography mass spectrometry approach in order to get the entire pool of detectable metabolites. And in this, each compound is represented by a peak on the mass spec, and the area underneath of that peak is correlated with the relative abundance of that compound. And to give you an idea of scale of how many compounds we can look at simultaneously with this method, we end up with, in my sample type, around 19,000 mass features or peaks, which could be potential compounds we could try to identify. But another approach is to look at a pared down list of compounds that we're interested in going into a study. And I'm going to refer to those as targeted metabolites. And again, for scale, this is around 88 compounds in my sample type. And specifically today, I'm going to be talking about a group of compounds referred to as compatible-solutes. And I'm interested here in how altering temperature and salinity conditions change the compatible solute pools in sea ice algae. And I'm interested in this because compatible solutes are these small organic molecules that can be maintained at very high intracellular concentrations, serve a number of roles inside a wide variety of cells, And two of the roles they can serve is mitigating salinity and temperature stress. And they can respond very rapidly to environmental change. So if we start with an algal cell out in the environment, in a marine environment, we would expect them to have a baseline concentration of compatible solutes in order to mitigate osmotic stress between the interior of the cell and their saline environment surrounding them. But if they're in colder or saltier conditions, like in sea ice brines, we would expect them to have higher concentrations of these compatible solutes to prevent water loss or freezing. And if they're introduced into warmer and fresher conditions, algal cells can rapidly dump these compatible solutes out into the surrounding environment. And once they're in the environment, they can be taken up and used as compatible solutes again by other organisms, used as a carbon, nitrogen, or energy source by heterotrophic bacteria in the sea ice, or serve as climate-active gas precursors. And here are just 3 examples of compatible solutes that have been found to be abundant in sea ice algae in particular, including DMSP, the climate-active gas precursor, glycine betaine, and proline. So to get at this question of how sea ice algae use compatible solutes in response to changes in temperature and salinity, we used a lab study of the Antarctic sea ice diatom Nichileconte, grown at 2 different temperatures and 2 different salinities, -1 and +4 degrees Celsius, and salinities of 32 and 41, and then compared this to environmental samples collected in the Arctic in Utqiagvik in a mixed sea ice community dominated by a niche species. And this was at similar conditions around -1 degrees Celsius and a salinity of 32. And first off, we could see from looking at the general physiology of the cells grown on these different treatments, there were relatively small changes in something like growth rate. So we saw less than 10% changes in growth rate, which is in stark contrast to a lot of studies of compatible solutes that use large shock treatments with bigger temperature and salinity changes, where growth often is reduced or stopped. So we're assuming that these cells are generally well adapted to the conditions we grew them under, but we still saw large changes in metabolite abundances, including those compatible solutes we're interested in. So just to give you a broad idea of the power of this metabolomics method, we wanted to first look at a suite of potential compatible solutes that we've seen evidence in other organisms that they could be compatible solutes. And here, this is again just this matrix of cold versus warm and salty versus fresh. And when we looked at this potential broad suite, we saw a number of reactions. We would expect compatible solutes to fall mainly up here, higher in abundance in the cold and salty treatment, but we saw some respond that way, and some respond only in higher abundance in just the cold treatment or just the salty treatment. Another group that responded either oppositely or had mixed reactions to what we'd expect for compatible solute action, and another group that were not significantly changed in response to any of the temperature or salinity treatments we used here. So we're seeing a really broad reaction to these conditions. And today I'm just going to talk a little bit more about a few of these compounds in particular. So those 3 compatible solutes I mentioned that are common in ice algae in particular, here I'm showing you proline, DMSP, and betaine. For proline, we saw that even though we were getting those less than 10% growth rate changes, really large magnitude changes in the concentration of proline, both in response to cold temperature and in response to higher salinity, with an additive effect up to a 4-fold increase in proline concentrations and reaching high concentrations around 50 millimolar. And here I have this as normalized peak area, which is like relative abundance, but we do have estimates for absolute concentration as well, which is that 50 millimolar. And then if we look at DMSP, again, we're looking at the normalized peak area against the salinities and the 2 different temperature treatments, -1 and +4. DMSP increased in response to cold temperature and higher salinity around 2-fold, but did not show the additive effect we saw for proline. And we don't have absolute concentrations for DMSP using this method, but we can assume from previous work they're similarly high. And for glycine betaine, they were at pretty high concentrations still, between 27 and 61 millimolar, but showed a more complicated response than what we're seeing for these other candidates, with a general increase in response to higher salinity, but a more complicated temperature story. And one of the cool things that this method can do is not only look at those compatible solutes we already know exist or are important in ice algal cells, but to find new potential candidates that act as compatible solutes. And one of these that came out of this study was DHPS, which is dihydroxypropane sulfonate. And this is a relatively newly studied metabolite in the oceans in general, and we don't have a great idea what it's doing in any ocean environment, let alone in sea ice, but it has shown potential as a compatible solute. And we are seeing similar results here, where DHPS was increased around twofold in response to cold temperature and to higher salinity, but again, not the additive effect we saw with proline, but reaching really high intracellular concentrations up to 91 millimolar. And just to give you an example to ground that, in a temperate diatom species, Thalassiocyrus pseudonana, DHPS is only found around 7 millimolar. So this is potentially a really potent compatible solute in the sea ice environment in particular. And the next thing, so the sea ice diatom nichulaconte maintains and regulates a diverse suite of compatible solutes with variable sensitivities to temperature and salinity, even when growth is not largely changed. But the next thing we wanted to look at is if the metabolome of this sea ice diatom culture was similar to that of a mixed sea ice community and what's actually going on in the environment. So here I'm just showing you the overall number of detected targeted metabolites from that smaller list I mentioned earlier. And this is for the field samples, which were a mixed community but dominated by a niche genera as well, Niche frigida, and comparing that to our nichulaconte culture. And we can see that the majority of metabolites in this targeted pool were shared, but there were unique compounds found in both sample types with more unique compounds found in the field. And the next thing I wanted to look at is how the concentrations of the compatible solutes in particular compare between these sample types. And here I'm just showing you for a select number of compatible solutes, the absolute concentrations for both the field and the culture. And the first thing I want to point out is the difference in y-axes, where the right axis in green is the millimoles of metabolite per mole carbon in the culture, and the right in blue is in the field. And you might notice that there is about a 10-fold difference in concentration between these two sample types, with concentrations being higher in the culture, which we would expect comparing a pure exponentially growing culture to a mixed field community where all the organic matter might not necessarily be living and growing. But overall, we saw that DHPS was the most abundant of the compatible solutes we quantified in both sample types, with betaine and proline scaling down in a similar pattern from that. But there were also a number of compatible solute candidates that were in relatively high concentrations in the field, like homarine and ethiopionic acid, that were not found in high levels within our culture samples. And another group of compatible solute candidates that were strictly unique to the field, not detected at all in our cultures, but at relatively low concentrations, hinting at some interesting possible species-specific differences in compatible solute use. And just to give you an idea of the scale of the concentrations of these compounds out in the environment and their potential to impact the environment, I want to remind you that these are highly mobile compounds that can be rapidly dumped from cells, such as during the spring melt when they're introduced into those warmer and fresher conditions. And they can dump up to 80% of their compatible solute inventory in less than an hour. So if we just take the nitrogen-containing compounds I have quantified here and pool them together, this is equivalent to around a 1.2 micromolar concentration of organic nitrogen that's capable of rapid efflux into the surrounding environment. And this is on a comparable scale to the inorganic nitrate concentrations in bottom sea ice during the spring, which are around 0.5 to 10 micromolar of nitrate. So this is a potential large source of nitrogen for this environment. So to wrap up, I want to conclude that metabolomics is a powerful tool that can detect a wide range of intracellular metabolites simultaneously in microbial organisms and quantify their response to varying environmental conditions. Here we saw that sea ice algae maintain and tightly regulate a complex suite of compatible solutes with variable sensitivities to temperature and salinity. And to follow up on this, we want to do continued work on environmental metabolomics of sea ice and compatible solute use in sea ice. And we're doing that with an NSF-funded project studying the spring melt season in Antarctica and seeing how that the sea ice community responds to melt conditions. So I'd like to thank everyone who is involved in this project, everyone who helped generate the data, particularly the Young, Ingalls, and Deming labs at UW, and I'll take any questions.
All right. Thank you very much, Hannah. We have time for one question, so there's a microphone up front and center. Please identify yourself when you ask your question. State your name and institution. Anybody have any questions for Hannah? Thank you.
Hi, my name is Joy. I'm at Carnegie Institute in Washington, D.C. Really great talk. Thank you for sharing your work. So you talked about the efflux of these metabolites containing nitrogen. Is there a way for you to understand the partitioning of extracellular versus intracellular metabolites?
With this method currently, it's a little difficult to look at the dissolved pool of compatible solutes, but that is something that the Ingalls lab is currently working on adjusting, and that's something that hopefully in the future we'll be able to quantify a little bit better. But currently we're just looking at the intracellular pool.
Thank you.
Thank you.